Mycoplasma Rapid Detection Kit Operation Guide. This protocol demonstrates a PCR-based workflow for Mycoplasma Rapid Detection Kit. PCR amplification culture the cells for about 48 to 72 hours, then harvest when they reach around 80% confluency. Thaw all kit components at 4 degrees Celsius for 2 hours or at room temperature for 30 minutes prior to use. Centrifuge at 1,000 revolutions per minute for 1 minute to collect the liquid at the bottom of the tube. Prepare 6 PCR tubes arranged into 3 groups with 2 parallel reactions per group. To compensate for pipetting loss, prepare a PCR reaction system for 8 reactions. In a 2 milliliter centrifuge tube, sequentially add TAC PCR master mix, Myco free water, and primer mix. Mix thoroughly by pipetting to prepare the PCR reaction mixture. Add 18 microliters of PCR reaction mixture to each PCR tube in the test, positive control and negative control groups. Add the corresponding samples to each PCR tube and mix gently by pipetting. Briefly centrifuge the PCR tubes. Place the PCR tubes in a thermal cycler and run the amplification using the preset program. After amplification, remove the PCR tubes. Analyze the PCR products by agarose gel electrophoresis. Gel electrophoresis analysis. Place the casting tray on a level surface and insert the comb into the designated slots. Add 67 milliliters of 0.5 times TBE buffer to a conical flask and add agarose to prepare a 1% to 1.5% agarose gel. Heat in a microwave for approximately 2 minutes until the agarose is completely dissolved. Add the nucleic acid stain and mix thoroughly. Slowly pour the solution into the casting tray to form a uniform gel layer. Allow the gel to solidify at room temperature for about 1 hour. Gently remove the comb vertically. Place the gel into the electrophoresis tank. Add 0.5 times TBE buffer until the gel is fully submerged. Load the PCR products into the wells, 10 microliters per well. Set the voltage to 160 volts and run electrophoresis for 15 minutes. Stop the electrophoresis and remove the gel. Capture images using a gel documentation system and save the results. Determine whether the samples contain mycoplasma contamination by comparing the results of the positive and negative controls. The expected amplicon size is approximately 500 base pairs with slight variation depending on the species.